Okay, so today's lecture, like I mentioned, is all about the muscular system. So this will be probably divided up into two parts. We're going to divide up kind of the first half, talking a little bit about muscle functionality, different types of skeletal muscle that we'll be discussing. And then we'll talk a little bit about how the neuromuscular junction is structured, how muscle fibers are oriented, what's the hierarchy of the anatomy of an actual organ of muscle, break it down into each of its individual components, and then we'll wrap up any material that we need to with our next lecture, where we talk a little bit about some exercise physiology, how muscles respond to fatigue, and then talk a little bit about different kinds of muscle contractions and some experimental approaches to how to test muscle behavior. All right. So let's talk a little bit about the functions of muscle, right? So we have three different kinds of muscle in our body. We have cardiac muscle, we have smooth muscle, and then we have skeletal muscle. Or if you're from Australia or England, skeletal muscle. So most of this chapter is going to be focused on skeletal muscle, all right? And that's distinct for a variety of different reasons. Skeletal muscle behaves differently than smooth or cardiac muscle. It's structured similarly to cardiac muscle, but again, there's some inherent anatomical differences there. But skeletal muscle is the most abundant in our body by like per unit mass. It has the greatest mass of muscle that we have in our body. So what the heck do we use muscle for? Well, one of the first ones is pretty obvious, is movement. Without muscle, we would just be these fleshy bags of bones and organs. We wouldn't be able to interact with our environment. So we've got muscle for that. We use muscle for a variety of different things, going along with movement, things like posture, things like production of body heat. So shivering, if you get too cold, we can do this really cool thing where we can decouple the neurological attachment to our muscles and contract and relax very rapidly so that we can produce body heat. So we'll also talk a little bit about why, when you shiver, why that actually produces heat when it may seem like it shouldn't. We have some specialized kinds of, smooth muscle that help with things like respiration, so ventilation of our lungs. We as mammals, and specifically humans, do this weird mechanism where we have to actively inhale or suck in air, and we don't have to think about exhaling. Other vertebrates, things like monitor lizards or even Komodo dragons, don't do that. They actually have to forcibly exhale, and then they passively inhale. So there's different kinds of breathing mechanisms that are related to respiration, but it's all facilitated by muscle. We use facial muscles for things like communication. We're one of the rare mammals that has facial nerve, and we have the ability to control facial expressions. Some of the other mammals that can do that are things like horses or other ungulates. So goats, weirdly enough, actually have the ability to communicate via facial expression, although very minor. Vasoconstriction, so we have the ability to change the shape of blood vessels or other vessels in our body, mostly blood vessels, but we can also do that with things like lymphatic vessels too. We can control their size and diameter by the activity of different kinds of muscle that are around them. And then ultimately, the reason we don't die when we go to sleep every night is because our heart is constantly contracting and forcing blood through our body, all controlled what's called autonomically, which is anatomy speak for automatically. So that's another thing that we'll end up discussing too, is that with muscle, there's kind of two different camps: muscles that we can control voluntarily, or we call voluntary muscle, and then ones that are controlled automatically without us having to think about it, which would be autonomic muscle. Skeletal muscle is under the voluntary camp. So we have complete and total control over our skeletal muscles and how they operate. We'll talk about muscle twitches and stuff later. But things like our smooth muscle and our cardiac muscle are all controlled automatically. You don't have to think about forcing food through your esophagus or through your stomach or your intestines. It does so automatically. You don't have to think about controlling how fast your heart is pumping, how much blood the heart is pumping out with each stroke. All of that is controlled automatically. So pretty cool stuff. So Yeah, like I mentioned, most of our body is skeletal muscle. So pretty much all of this muscle that's attached to our bones is going to be considered skeletal muscle. It's got a distinct microstructure that we'll look at here momentarily that's going to separate it from things like smooth muscle and cardiac muscle. And smooth muscle is typically found, around most of the vasculature in our body. So most arteries and veins are surrounded by or have a component of smooth muscle. Generally, the bigger the vessel, the more smooth muscle there is. And then we have things like our intestines that are surrounded by smooth muscle. We've got our stomach that has smooth muscle, the esophagus as well. All of those main structures that are responsible for transmitting food from our mouth through our rectum are all going to contain smooth muscle. And then cardiac muscle is found only in the heart, right? So things about muscle, one of the main things to understand, and you'll see this kind of pop up, and so Shania, you brought up a really good point earlier about some, terminology that can be, used in certain ways. Sometimes I'll throw terms out there that you may not find in your book or in the lab manual for that matter. But one thing to be aware of is that what you'll probably see written or printed in your book or in the lab manual, and this is what you should go by, but it's more of just a something for you to think about, is that muscle has various kinds of actions that they can perform depending on what they're attached to, and when they contract, what they end up doing. But the only function that a muscle has is contraction. And what you'll probably see written is depending on where a muscle is attached, it'll have the function of like bending the elbow or flexing the knee. So you can go ahead and use terms like function to describe what different muscles are doing. But understand that, if you guys decide to go into healthcare or, you decide to go farther in anatomy, it's commonplace to think about the activities of muscle as actions depending on where they're attached in our body. But the only function that whether it's skeletal, smooth, or cardiac muscle has is to contract. Okay. So whenever a muscle contracts, all it is shortening. So we've got a series of overlapping protein fibers called actin and myosin that when they're stimulated will increase the amount of overlap and they'll cause a contraction. So if you flex your arms or things like that, you're performing muscle contractions. Anytime you exercise, lift weights, run, do anything like that, you are performing muscle contractions. Okay. But depending on where they're attached in our body, that determines the different, the different actions that they do. There's this other interesting thing called excitability. So muscles are excitable. That means when they're exposed to something called an action potential, which is generally a neurological stimulus. Sometimes it can be a chemical stimulus specifically, but sometimes it's either electrical or chemical, and that'll cause some sort of reaction in the muscle. In general, those are what we call excitability or excitatory postsynaptic potentials, or EPSPs. But sometimes we can actually inhibit the function of muscle. We call those inhibitory postsynaptic potentials, or IPSPs. But we'll get to those later on when we talk about the nervous system and how that integrates with muscle. But so all muscles are excitable. Once they're stimulated, they can do something. And in general, that something is a contraction. Now, I know I've just said a bunch of times that the only thing muscle can do is contract, but passively, when muscle isn't being activated, it can extend. We can stretch out our muscles, right? So this is exemplified every time, as you're getting ready to work out, you might stretch your muscles, right? Or if you've been sitting at the keyboard all day and you do your forearm stretches or things like that, right? So, we can stretch out our muscle, and our muscle has, varying degrees of compliance that's associated with them. So stretchiness. Depending on how much muscle mass we have in a given muscle that we're stretching will determine that extensibility. But all muscles have some amount of stretchiness or extensibility that's in there. So extensibility and elasticity is kind of the opposite of extensibility. Is the muscle able to resume its original resting length? So extensibility and elasticity look similar, but are not technically the same thing. Elasticity is how easily can this tissue resume its original structure, its original starting length after we've stretched it out. And in general, muscle is pretty good about doing that with the help of things like tendons and ligaments that can assist in that process. But yeah, so those are some of the general properties of muscle. The only function of muscle is to contract, but depending on where it's attached, we can have various kinds of actions that muscles will perform. So here we've got a nice little table. So table 7.1 from your text that breaks down each of our different categories of muscle. So on the left-hand side, we have skeletal muscle. Where do you find it? Attached to bones, hence the skeletal muscle name. Appearance, long and cylindrical. They have these little tiny banding patterns called striations, or it gives it a striated appearance. Those areas where we have dense pockets of what we call myosin, or it's a heavier protein, that it's all interconnected with our actin filaments. So what you'll find in skeletal muscle is that there's a ton of these striation patterns, but they're all nicely laid out in a really rigid formation. It's pretty cool. The other cool thing about skeletal muscle is that they are multinucleate. Most of the muscle mass of a cell, which again, for a muscle cell, it's one of these tubes right here. That is a muscle cell. Most of the volume of a skeletal muscle cell is protein. It's going to be mostly actin and myosin. And if we're going to produce a bunch of protein, as you recall from chapter 3, what we need to do is we need to have a ton of nuclei because that's going to be carrying out all the information for protein synthesis. So skeletal muscle are generally multinucleate. Sometimes in really long muscle fibers, like we have in some of our leg muscles, we can have hundreds of nuclei in a given muscle cell. They're really, really dense in their nuclei because we need to produce a lot of protein to satisfy their function. Okay. These skeletal muscle cells do not have any particular intercellular connections. We'll discuss that a little bit later, probably with the next lecture. I'm going to skip over the smooth and cardiac muscle for now, just because of time constraints. But what you'll find is that smooth and cardiac muscle have special intercellular connections that are going to be relevant for how they function. Striations, yes. Autorhythmic, no. So these are not controlled automatically. So as much as I don't know if any of you guys grew up with siblings, but as much as you would try and pass off like smacking your sibling upside the head, like, oh, I couldn't do it, I didn't control it. My body just acted of its own accord. Or is it just me with my younger brother? I had younger brothers 5 years younger than me, and we would always rough and tumble like that. But I was never able to pass off my body acting of its own accord. So our skeletal muscle is always voluntarily controlled. Okay. Function, move the whole body, but the main function is to contract. Depending on where it's attached, we can have various kinds of actions. So go ahead and take some time on your own to go over some of the structure of smooth and cardiac muscle. They vary significantly from skeletal muscle, especially in appearance. And again, as we get into those other chapters, we can go into some more detail, or if we have some time at the end of our next lecture, we'll discuss that as well. All right, so one of the nice things about muscle, whether it's smooth, whether it's cardiac, whether it's skeletal, is they're kind of all similar in the way that they are all pretty much just tubes, all right? So an individual muscle fiber, which would be the muscle cell, okay, that can contain tons and tons of what we call myofibrils and then myofilaments, but we'll get to that in a little bit. But all muscle is really just tubes within tubes within tubes. And these tubes will shorten when they're activated and then extend or lengthen when they're relaxed to get back to their original length. And then sometimes they can be stretched out, all right? But that's generally how an entire muscle organ is put together. It's just you put tubes within tubes within tubes. So if you look up here on the top left, we have our ultrastructure of an entire muscle organ. So like take your biceps brachii muscle right here. So the main one, like if you were like, oh, flex your muscles, that's the one we would all do, right? So the biceps brachii, when it's all put together, has this outer connective tissue called the epimysium. Now, we've seen that term before when we were talking about bone, when we talked about epiosteum, or at that point we called it the periosteum because the ultrastructure of bone is not as rigorously put together as muscle is. But we'll get to perimysium here in a little bit. But so this epimysium out here can be broken down into muscle fascicles, okay? Each of these fascicles here is going to contain multitudes of muscle fibers. Now remember, the muscle fibers are a muscle cell, okay? So we can wrap up a bunch of these muscle cells next to one another inside of this purple connective tissue right here, which is what we call the perimysium, okay? And then we have our individual muscle fibers, which is again our muscle cell, that are surrounded by the endomysium. So we have epi, peri, and endomysium. So epi meaning outer, peri on the periphery, and then endo meaning innermost layers of connective tissue surrounding muscle. So that word my or myo, which is what it comes from, is equivalent to or means that we're talking about muscle. Okay. So again, as I mentioned, inside of all of our skeletal muscle fibers right here, it's just all protein. All of it is protein. So we've got a ton of these, what we call myofibrils, which are these big green tubes that we see here. Excuse me. And these green tubes are a cross section through a series of proteins that are all interconnected with one another. And those, what we call contractile proteins or myofilaments, are actin and myosin. All right. So the thing to note about muscles is that the terminology is really obnoxious to get a handle on. So this is just going to take some time of, writing it down, writing down the hierarchy, being able to understand what each of these layers are. It's just one of those things that takes time. But the nice thing about muscle fibers, and again, it's the same thing with neural fibers that we'll talk about on Thursday a little bit, is that they have all the same, most of the same internal cellular components as most of the other cells in our body. So they all have, endoplasmic reticula, they've got mitochondria, they have smooth endoplasmic reticulum, which in the case of our muscle cells just gets a fancy name called the sarcoplasmic reticulum, which its main goal is to store a bunch of calcium, which is sort of similar to the smooth ER that we find in kind of a traditional eukaryotic cell. But this yellow stuff just basically provides a meshwork bed that holds a bunch of calcium. And as we'll find, calcium is going to be very important for muscle activity. Just as we had talked about at the beginning of the year, calcium is one of our most important ions that we have roaming around our body. We use it for a bunch of different things, and a lot of it has to do with cell-to-cell communication and cell activity. So for example, here with muscles, what we'll find is that these myofilaments really only respond when calcium is involved. All right. So this big yellow sac, the sarcoplasmic reticulum, releases calcium when it's stimulated. The membrane of our muscle cell is called the sarcolemma. The cytoplasm of a muscle cell is called the sarcoplasm, because it's all referring to the functional unit of the muscle cell, which is a sarcomere. Okay, so a lot of that terminology that you guys will see here is going to have things like myo referring to muscle, or sarco referring specifically to the components of the muscle activity, ending and then restarting automatically. Is that happening to you guys, or just you, Chris?
Yeah, well, to me, it's like pretty much the call just ends and it goes to the home screen of Zoom, and then it automatically connects back to you. But it sounds like you keep talking, so I'm not sure if anyone else is dealing with it.
Huh. That was actually what was happening to me before when my internet connection kept dropping. So Chris, what might work for you is if you like either restart your router or restart your computer, and then hopefully things work out. So if you want to do that, if it keeps doing that to you, I would recommend trying that.
Okay, yeah, I'll just drop out of it, fix that, and come back.
Okay, cool. Okay, so this is how our muscle cells are put together. At their functional level, we've got these two little proteins called actin and myosin that overlap one another. And when they increase that degree of overlap, that's called a contraction. And then when our muscle is attached to something, it pulls on it. And then we're able to produce force and interact with our environment. So let's take another look at what a skeletal muscle looks like under a microscope. So in the top right here, this is a skeletal muscle, probably around 100 magnification, or it might even be a little bit less than that. But we can see we've got all these little concentric rows of our little overlaps of actin and myosin here. So each of these bands right here, these dark bands, okay, the A band, that is where we have our overlap of actin and myosin. The dark purple middle component is, annoyingly enough, actually indicative of the green protein that we're seeing here, which is myosin. So myosin is kind of our main attractor of actin. So this is what contains the pulling force that's produced when a muscle contracts and is stimulated. So our myosin molecules, excuse me, have these little rods with these head portions here. These head portions kind of act like little ratchets. When they're stimulated, they pull on the actin filaments here and pull these fibers closer towards this midline called the M line. When that happens, that's what a muscle contraction is. And then when there's not calcium involved, because again, calcium is going to be our signaling guy here, then the actin filaments will relax, and then the whole sarcomere, which is what we're seeing right here, separated or denoted by each of our Z discs right here. So these little dark single lines right here, those denote the end of a given sarcomere. So all of the stuff in between each of our Z discs, that is a given sarcomere, okay? So really, what determines muscle health and muscle activity, if you're exercising and let's say you're doing bicep curls, why is it a lot easier to do a bicep curl when you have the weight, super close up here and you only do that? Why is that easier to do a bicep curl than when you have your arm fully extended out and then you do a muscle contraction? A lot of that has to do with some basic physics, but it also has to do with the length that our muscles are operating at. When our arms are fully extended out, we're actually stretching these actin filaments a little bit farther than what they usually like to be at. So it's much harder for us to produce a lot of force if our arm is fully locked out than it is if we have our arm at a 90-degree angle and then we activate our muscles. So, our muscles are sensitive to whatever resting length the sarcomere is sitting at. So if our muscles are super stretched out, these sarcomeres are kind of, pulled apart a little bit more. Another way to think about that is the myosin heads right here do not have a lot of overlap and don't have a lot of attachments with the actin filaments here. Okay. So kind of backing up a little bit, let's talk about some of these zones and bands here. So we've already discussed the M line right here, which is the central portion of where our myosin filaments are set up. We have this zone called the H zone between each of the ends of our actin filaments on either side of the sarcomere. This H zone and the size of it will determine whether or not the muscle is contracted or relaxed. If the H zone is relatively large, then the muscle is either at rest or it's being stretched. And then if the H zone is quite small, almost non-existent, then that's indicative of the muscle being contracted. Okay. The A band tells us the overall length of our myosin fibers. And this can vary depending on animal. It can vary depending on what muscle you're talking about in the body. Also, the darkness of it or the density of the A band will also tell you a little bit about some of the identity of the myosin proteins here that can make up a given muscle fiber. And then we've got the I bands, which separate the ends of one set of myosin filaments with the ends of another set of myosin filaments in a neighboring sarcomere. And then we've got this Z disc. The Z disc is a little sequence of collagen and protein that helps route the actin filaments into these locations so that when the myosin heads pull the actin filaments towards this M line, then the actin filaments will stay rooted in the Z disc, and then the overall sarcomere will shorten as a result. All right, let's see if I'm missing anything right here. I don't think so. Okay, so we've already talked about myosin. This is a very simplified version of what myosin does and how it operates. But interestingly enough, the more regulatory protein of this entire process is actin. So actin has this really interesting setup where we have these little purple beads. This is called G-actin or globular actin. And they have these little active sites that are noted or demarcated in yellow right here. So those active sites at rest are protected by this purple string called tropomyosin. That tropomyosin is important because it makes sure that let's get Chris back in here. The tropomyosin makes sure that the actin filaments aren't just being willy-nilly attached to myosin and that our muscles aren't just constantly being contracted, right? Because if the tropomyosin wasn't there, then these active sites would be constantly exposed, and then the myosin heads would be constantly pulling our muscle fibers towards themselves or towards the M line. So what that would look like is you would have something like rigor mortis occur. So post-mortem, a condition called rigor mortis can occur or will occur when the body cycles through all of its ATP and calcium and can't keep the tropomyosin complex covering these active sites. And then the muscles themselves will end up contracting and seizing, okay? So then we have this tropomyosin fibrous protein, and then we have a troponin sequence of proteins as well. These troponin molecules will help make sure that the tropomyosin stays covering our active sites. And with the inclusion of calcium into this matrix, then we change the shape of the troponin, which then in turn removes the tropomyosin away from the active sites. And then our myosin heads can then attach to those actin filaments and then create what we call a cross bridge. And that cross bridge cycle is essentially what we do when we, flex our muscle and then we relax our muscle. Okay, so we're going to get to that here in a little bit, but that's essentially the entire process of muscle contraction and muscle relaxation. All right, so let me just get my bearings here. Let's talk a little bit about what we call the neuromuscular junction. This is kind of cool because it previews what we're going to discuss next chapter when we start talking about nerves and neurons and how what we call action potentials operate. Okay, actually, before we get going, I know I've kind of spat a lot of terminology at you guys in the past couple slides. Does anybody need me to clarify anything? Or does anybody have a question about a term that I used or need clarification on something before we move forward? I also want a sip of coffee. Yeah, Mazen, absolutely. So the Z disc is this little blue guy right here. So it's a combination of collagen and protein that helps keep the actin fiber. So this actin protein here, it's kind of like a wall. You can think of it that way. The actin filament is rooted or attached to that wall so that when stimulated, the entire sarcomere will shorten because those walls are getting closer and closer to this M line. It'll make a little bit more sense. I've got some other pictures in here, or if you flip ahead in the textbook, if you have it with you, you'll see here, let me just see what that figure is called. So it's this figure right here. So figure 7.7, Mazen, if you want to look ahead at that really quick, that'll help explain a little bit about what's going on with the Z disc and why that's important, because it's essentially the root of where the actin filaments are attached to the ends of the sarcomere. What's not depicted here, but there's also a coiled spring, which is a protein called titin that attaches to this little indentation where the Z disc is in the end of the myosin, which essentially just helps the muscle resume its original resting length. There's some pretty cool folks over at Northern Arizona University that look at what happens when you genetically modify muscle to not express the gene to create that titin protein. And they've done some really interesting research about muscle activity and behavior in some mice models, which is kind of cool. But yeah, so the Z disc, that's essentially what the Z disc is. It's the end walls of the sarcomere, and the Z disc length or the distance between them will change. It'll get shorter when the muscle is contracted. It'll stretch out when the muscle reduces or resumes its original length or is stretched. Okay, so hopefully that kind of clarifies that. Okay, so that's a good question. Anything? Okay, cool, good. Anybody else have a question before we move forward? All right. Well, if you think of something, throw it out there. So now this next figure is talking about the excitability of muscle fibers. Okay, so how do we activate these things? Or how do muscles respond to external stimuli? Because what's kind of interesting to think about too is we can inject things into our muscles to determine what they do or to determine what they don't do. We have different kinds of poisons that can affect muscle in both an excitatory or inhibitory way. So we have, different kinds of paralysis that can come about. Um, so muscle fibers by nature, since they are cells, are what we call excitable. They have membrane channels embedded within their membrane, but they can let in various kinds of ions, things like sodium or potassium, and that will determine the activity or the excitability of that muscle fiber. So in general, when we talk about excitability, we're referring to like a positive stimulus or something that increases activity. Whereas inhibition will typically lower the activity or lower the response of that muscle. I'll give you some examples here in a little bit when we talk about neurotransmitters. But so all skeletal muscles have this thing called a resting membrane potential. So Shania, I don't know if you have your study guide open or if any of you guys are contributing to that as you go along, but a good point to throw on that study guide that I would recommend is being able to define what a resting membrane potential is. So like it says here in part 1, sodium channels in pink and some, but not all, potassium channels in purple are closed. Potassium diffuses down its concentration gradient through the open potassium channels, making the inside more positive, or the inside of the cell membrane negatively charged compared to the outside. So in general, this is another thing to recognize too. This idea of a resting membrane potential factors in two things. Factors in the chemical gradient. So the movement of ions down their own concentration gradients. It also factors in the electrical gradient. So which side is positive and which side is negative. Together, we call that an electrochemical gradient, which is very similar to our membrane potential or resting membrane potential. Okay, so how this thing changes, how our resting membrane potential changes, is going to determine the activity of whatever structure we're talking about, in this case, a muscle fiber. And I want to point out the word potential because sometimes I know that this trips people up sometimes. The word potential is referring to the potential to transform energy from potential energy into kinetic energy. That's the whole premise there. So when we talk about a resting membrane potential, what we're talking about is at rest, the muscle cell is prepared to perform some sort of work. It's prepared to transform the potential energy that it has stored up outside of the cell, where we have lots of sodium, and it's prepared to open up these channels and allow sodium to flow into the cell and move down its gradient, thereby transforming that potential energy into chemical energy. So that's where that term potential comes from. So hopefully that stands out to you. Define potential. So I'm throwing that out there because that's an important term to recognize, because moving forward, we'll discuss action potentials, which are essentially going to be, using this electrochemical gradient, but in nerves, to generate electricity and create electrical currents. Okay, so resting membrane potential, should know what that is, be able to define that. And then we have the next stage where we have the transformation of that potential energy in the form of our artificial concentration gradients into kinetic energy. And the fancy word for that is called depolarization, where the polarized nature of the membrane will then become depolarized, or it'll flip. Okay, so we can see here that as we allow sodium ions to diffuse into the cell, what was once negatively charged inside becomes positively charged because all of our sodium ions are positively charged. So we just add more positive stuff in here. We make the inside positively charged. The outside becomes negatively charged. That's what we call depolarization. So then in order to reset this, because a depolarizing event is usually indicative of some sort of activity that's going on, but if the cell remains depolarized, it's not going to be able to respond to any other kind of stimuli. So we've got to be able to reset that gradient. So we've got things like the sodium-potassium pump, which will shove sodium ions back outside, cram more potassium ions back inside. We've also got these leaky channels that can let some of them passively flow down the gradients if it gets too well established. But then ultimately we reset the membrane and we're ready to go again, and we're ready to respond to another stimuli. So at rest, all of our cells of most of the membranes in our body have a positive extracellular charge and a negative internal cellular charge. Okay. Upon stimulus, and we'll talk about the different kinds of stimuli momentarily, we can transform that membrane potential, the extracellular potential from the outside, from positive into negative. And by definition, we flip that polarization. All right. So understanding this idea of resting membrane potential. Let's see. Hold on. Can you guys hear me still? Okay. Sorry. Okay, cool. My mouse stopped moving and I was like, oh my gosh, what's going on? But yeah, so recognizing this idea of membrane potential, depolarization, repolarization, this is going to be key moving forward because all of the other components or the physiological mechanisms we'll discuss respond to resting membrane potential. If a cell is going to respond to some sort of a stimulus, usually from the nervous system, it's got to be able to undergo this depolarization, repolarization mechanism. Okay, cool. So kind of leading into how muscles know when to contract and when to stay relaxed is we have attachments to the nervous system. The nervous system pretty much controls everything that goes on in our body. There's an argument that hormones have greater control over our body, and I would say like long-term changes, absolutely. But on the day-to-day, immediate control of everything that's going on in our body on a constant basis, the nervous system is bar none, controls everything. Because every single one of the cells in our body is going to have connections via these nerve fibers. These nerve fibers have these little, what's the term? Presynaptic terminals. They're also, the actual term for this structure right here is called a telodendrite. You can refer to it as a presynaptic terminal for now, but once we get to the nervous system on Thursday, you'll learn presynaptic terminal, or I'm sorry, telodendrite, because the ends of these house different kinds of chemicals. And most commonly, the chemical that's housed in the ends of our nerves that attach to either a muscle or some other kind of organ is going to be called acetylcholine. So let me put that in the chat here. So it's called acetylcholine, commonly abbreviated ACH. So you pronounce that acetylcholine. And so this particular neurotransmitter is a chemical that when it's released will cause a depolarizing event on the membrane of our target cell, in this case, a muscle cell, which then will in turn transmit that signal into the muscle fiber, into the myofilaments, and then cause a contraction to occur. So here is what that looks like underneath the microscope. So we've got our little nerve ending right here, neuromuscular junction, where we have each of our little presynaptic terminals, aka telodendrites, that are going to then be attached to our muscle fiber right here. So while the actual nerve might be attached via various kinds of protein extensions and attachments, the actual presynaptic terminals do not have a direct attachment to the opposing membrane over here. Okay, they're not physically attached. There's actually a space in between these structures, so in between this structure and this structure. All right. Also notice the folded membrane here, so to increase surface area to volume ratio. All right. So now let's talk about how muscles operate. Okay, so this figure gives us a lot of information kind of all at once here. But what it's telling us is that we've got our nerve that sends an electrical signal in the form of an action potential, and it reaches that presynaptic terminal. Those presynaptic terminals will first respond to the action potential by opening up calcium channels. Okay, so one of the first instances, and definitely not the last, of the importance of calcium. Calcium will then enter the end of the neuron here, and then it'll trigger this process called exocytosis, where these little synaptic vesicles that contain the particular neurotransmitter are then going to be released into the synaptic cleft. So these neurotransmitters will then meld, or I'm sorry, the vesicles here will then meld with the membrane here and then expel their contents into this cleft. And then via diffusion, so moving from high to low concentration, these chemicals will then move from the presynaptic terminal over here and then attach to these protein receptors on the target structure, on the target cell. In this case, these are kinds of proteins that we call chemically gated channels, or sometimes called ligand gated channels or ligand activated. Ligand is just a fancy word for a specific chemical that activates something or inhibits something. So these particular protein channels here will then open up in response to acetylcholine binding to it. If there's no acetylcholine, then they don't do anything. Only when acetylcholine is present and attached to it do they open up. When these protein channels open, they will allow for sodium to flow into our cell over here and then cause the postsynaptic membrane response. In this case, it's an excitatory response. That action potential or electrical signal, think of it as like a go signal. Like when you're telling your muscles to contract, this is one of the first things that happens between your brain and your muscle. It activates our sodium gated ligand channels here with acetylcholine, and then they allow sodium to rush in to the membrane or into the inside of the cell to cause that depolarizing response. Okay, and then ultimately the acetylcholine gets released and is broken down and recycled and then reintroduced back into here. Then we recreate acetylcholine and then it's ready to go again. Right. So let's see. Yeah, so I had mentioned we have various kinds of proteins that can inhibit the activity of acetylcholine. Some things like organophosphates that will actually inhibit the activity of something called acetylcholine esterase, which is the enzyme that breaks down acetylcholine. Because as it turns out, acetylcholine and most what we call neurotransmitters don't really stay bound to these proteins for a long period of time because then if they did, they would cause much longer lasting effects. We do have chemicals that do that in our body. We call them neuromodulators. So things like serotonin and dopamine are examples of neuromodulators that have much longer lasting effects on the target structure. And then we have various kinds of narcotics and drugs that can mimic the effect of neuromodulators and outcompete them for the proteins that they activate, and then cause much longer lasting responses in our body with those various kinds of euphoric sensations. But sometimes if we can inhibit this acetylcholine esterase, then we can effectively eliminate the body's ability to break down and recycle acetylcholine, and that can have adverse effects on the target cell. Like a muscle, for example, will cause something like contraction without relaxation or spastic paralysis, right? Then we can have other kinds of nasty poisons that will bind to acetylcholine receptors and outcompete them and cause something called flaccid paralysis. Whereas instead of causing a excitatory response in the muscle, we inhibit it and we decrease the activity of the particular structure. So that would be an example of what we call an inhibitory postsynaptic potential, or the IPSP. So differences between different kinds of paralysis: flaccid paralysis versus spastic paralysis. All right. Cool, we're making good time here. This is good. So what does this look like in muscle? Because that's what we care about right now. What's going on in the muscle fibers when we have all of these acetylcholine molecules do all this stuff? Well, that action potential gets transmitted downstream and then will ultimately cause the sarcomere to contract. So here, with our relaxed sarcomere, we have a relatively wide H zone right here. So the space between each of our actin filaments on either side. That space is relatively wide. We have this little spring-like protein called titin that's depicted right here, and it's relatively relaxed. It's not compressed at all. The A band itself is relatively small, and the I band size is quite large. But then when we contract, we can see that that changes. So our titin proteins get all coiled up and compressed, very small to non-existent H zone. I bands are contracted, and then the A band itself remains quite large. Okay. All right. So this is our contracted sarcomere, and then we have our relaxed sarcomere. So really notice how those actin filaments come closer together. So what we'll see is these myosin heads will essentially walk their way towards the end of the actin filaments as they're rooted in the Z disc right here. But then they can resume that original length with the help of this titin protein that helps kind of expand and push them out. Okay. So how the heck does what we just talked about happen? So we've got our excitatory postsynaptic potential over here with the help of our action potential, acetylcholine, triggering that depolarized response in our muscle cell membrane. That signal of depolarization essentially hops its way along the cell membrane here and will then ultimately reach the sarcoplasmic reticulum, which remember is just this giant sac that also holds a bunch of calcium inside of the muscle cell. The sarcoplasmic reticulum is tightly integrated with the sarcomere so that when it's stimulated, it will release calcium ions that are really close by proxy or by proximity to the myofilaments and then cause the myosin heads to attach to actin and pull the actin heads towards that center M line, creating a muscle contraction. So with the addition of calcium from the sarcoplasmic reticulum here, that calcium transforms the shape of the troponin-tropomyosin complex on actin. So the calcium binds to troponin. The troponin causes a physical change in the shape of the tropomyosin protein that's normally protecting all of these active sites. Those active sites become exposed, then the myosin head attaches onto them and then pulls them forward towards that M line right there. And then with the help of some ATP, we can release them, reset their resting length, and allow for the the sarcomere to relax and resume its original resting length. Okay. So that's a very condensed version of the next thing that we'll discuss here. And then once we get through this, this next thing called the cross-bridge cycle, then we'll actually end for today. All right, so the cross-bridge cycle is important because it determines how our muscles produce force, right? And as of right now, I believe this is still technically what we call a theory. And I'm going to put that in quotes because recognizing that most scientific discoveries are recognized as, or scientific hypotheses or theories are recognized as such because they have a lot of evidence to suggest that this is actually the way that they operate. But to my knowledge, I don't think that this has yet been observed to actually be happening in a muscle cell. But with what we know about how these proteins are structured and configured and what we've seen of their ultrastructure, this is probably how everything works. But keep that in mind. That's kind of an anecdotal thing. You won't be tested about whether or not this is fact or theory or anything like that. But recognizing that the cross-bridge cycle, or what we call the sliding filament model, is, as of right now, I believe, still technically a theory, but it's backed up by a lot of evidence. So really, what this cycle represents broken down is the detachment of myosin and actin. The attachment, the pulling of the actin filament towards the midline, what we call the power stroke, and then the ultimate relaxation of myosin, the resetting of the actin filament, and then if the muscle is still stimulated, the ultimate attachment of the myosin head again, pulling that actin filament towards the midline. So let's kind of break that down and look at what that looks like up close. So at rest, okay, these myosin heads and actin filaments are not attached, all right? So they're not attached to one another yet. But if we introduce calcium, okay, calcium will then cause that change to expose those active sites. When those active sites are exposed, we end up creating what we call a cross bridge. So the cross bridge here is where we have the myosin head attached to the active site of the actin filament. That's where we create the cross bridge. Once that cross bridge has formed, then we have a physical change in the shape of the myosin head by pulling that actin filament backwards, okay, or towards the midline. So the myosin heads will bind to the exposed active sites on the active myofilaments to form cross bridges. And phosphates are released from the myosin heads as we change that configuration of ATP to ADP. So next up, we have what's called the power stroke. Energy stored in the myosin heads due to its physical orientation, it's another form of potential energy, will spring the myosin head forward. So energy stored in the myosin heads is used to move the myosin heads, the small dark blue arrow right here, causing the actin filaments to slide past the myosin filaments, the dark blue arrow up here. And ADP molecules are then released from the myosin head. So then the ATP finally gets released there. All right. So the myosin heads are pulling actin towards that midline. Crossbridge then releases once that power stroke has been performed. ATP molecule binds to each of the myosin heads, causing them to detach from actin. That ATP gets quickly broken down into ADP and phosphate, which remain attached to the myosin heads, helping keep it helping keep it separate from the actin filaments there. Okay, and then we've got our recovery stroke, where the heads of the myosin molecules return to their resting position, so small dark blue arrow, okay, and energy is stored in the heads of the myosin molecules. If calcium is still present, okay, so if it's still attached to the tropin and tropomyosin complex, then we can go through this whole cycle again, where myosin will then attach to the active sites, create a cross bridge, initiate the power stroke, and then pull that actin filament towards the midline. Okay, so you can also think of that as like, if you're lifting something and you're holding something up, you're constantly contracting your muscles, whether you think about it or not. We're constantly creating cross bridges, and we're constantly having those myosin heads keep actin close towards that M line of the body, or the M line of the myosin body. And then as we increase muscle fatigue, then we're unable to continuously recycle that and perform that power stroke because of buildup of other proteins and other chemicals in the muscle that prevent this process from happening. Okay. Usually it's a deficit of ATP in the muscle cell, right? Or the size of the muscle fiber will also contribute to that too. All right. Let me see. Let me see here. You know, all right. We're almost done. I might just finish this off. Actually, no. No, we'll finish this off on Thursday. This will only take like 15, 20 minutes of class time on Thursday. I'd rather not just like shove this down your guys' throat right now. So we're going to stop here for today. Okay. So this will be the end of part 1. We'll finish this up with our next lecture.